Chapter 3 is dealing with the cell structure and function. So we're going to look at different cell types, the different structures within the cells, what their function is, etc. There are certain characteristics or properties that are unique to all living organisms, and we use these basically. If you have these properties, then you'll be classified as a living organism. Some of these things are growth, reproduction, responsiveness, and metabolism. And in this table, we look at these things like growth. It's the increase in size. That's going to occur in everything such as bacteria, the archaea, the eukaryotes. And so this is comparing that viruses do not increase in size. Whatever size they are initially, that's the size they do remain. In terms of reproduction, you're talking about the increase in the number of cells, not the increase in size of the individual cell, but the number of them. And this also occurs in all organisms. Viruses, however, can only reproduce inside of a host cell. They are dependent upon that. Responsiveness is how do you respond to various stimuli that are present in your surrounding environment. Metabolism is the reactions, the chemical reactions that are occurring in the organisms that are necessary for its survival. In this case, all organisms are capable of carrying out metabolic processes. Viruses do carry out some, but they are also very good at utilizing their host cells' metabolism. And then the cellular structure deals with the structures within the cell that help them to take be able to help them carry out or take part in all of these various processes. Now, viruses don't have what we refer to as a cellular structure. They do have a structure to them in that they tend to have a capsule or a coat and that surrounds nucleic acid, but they don't have cytoplasm within them. Cells occur in all different shapes and sizes. Here are just a few examples of them. In the photograph marked A, you do see this is from a scanning electron micrograph, and the size scale there is 1 micrometer. In microbiology, a micrometer we often refer to as a micron. But you can see there are these small little rods, numerous of them there. LM in figure B stands for light microscope, and you can see here now the scale is very different. It's 40 micrometers. So the cell on B is much, much larger than that on A. Cells are going to be divided as either prokaryotic or eukaryotic. We're going to look at the differences and similarities between them. So we're going to start with the prokaryotes. The prokaryotes are the simpler, smaller type of cells. From an evolutionary standpoint, they are the ones that have been here longer. They do not have a nucleus. Now, they do have DNA. It's just not in a nucleus. Because they don't have a nucleus, they can read the DNA and make protein at the same time. You cannot do this with cells that have a nucleus. They do not have membrane-bound organelles within the cell. When you see these with eukaryotic cells, the function of these specialized organelles, a prokaryotic cell can still carry out those functions, they just do it a little bit differently. They tend to, as I said, to be very small. Your bacteria and your archaeas are the two groups that comprise the prokaryotes. In this representative drawing, you do see the typical prokaryotic cell that the nucleoid, that's just a region where the DNA tends to accumulate in, but it's not a nucleus. You do have the cytoplasm, which is the fluid inside the cell. You do have ribosomes that are present. You do have various, depending on the cell, various inclusion bodies. You may have these little vesicles with maybe iron in them, maybe pigmentation. It just depends on the particular species you are talking about. Some may have a flagellum, some may not. They will have cytoplasmic membrane. Some of them may have a cell wall. They may have an extra layer beyond that as well. Eukaryotes, they do have a nucleus. They do have internal membrane bound organelles that will have very specific functions. They tend to be much larger than your prokaryotic cells. So they're larger, they're more complex. They're the new kids on the block, if you will. This would include things such as your plant cells, your animal cells, protozoans, algae, and fungi. And this is a representative drawing of a eukaryotic cell. Just as the previous picture of a cell, part of it is actually a TEM, which is a transmission electron micrograph that allows you to see the more three-dimensional internal structures, and then the schematic drawing added to it. So you can see that there is the nucleus. The DNA is going to be found inside the nucleus. You're also going to have another structure inside the nucleus known as the nucleolus. And then outside the nucleus, you're going to have other structures like ribosomes. You have these membrane-bound organelles such as the Golgi body, the endoplasmic reticulum, of which there's both the smooth and rough, and other vesicles. You do have the cytoplasmic membrane, just like you did with the prokaryotic cells. There may or may not be a cell wall, depending on what type of cell you're talking about. In terms of sizes, like I said earlier, they come in all shapes and sizes. In this picture, you do have the approximate size of a chicken egg. A chicken egg is considered a cell, one cell, very large. Now, as you can see, that little dot on there is representative that if you enlarge that, you can see things like a protozoan, Giardia. That is the protozoan that's often in water that makes you sick. And then comparative size to that would be a bacteria, which is Staphylococcus. It's on most people's skin, on a lot of surfaces. If it penetrates in the body, it can cause different types of infections, such as food poisoning, skin rashes, etc. And then there's also a virus. So you can see viruses are the smallest of the organisms that we are going to be talking about. And bacteria are huge as compared to a virus, but they are actually very small compared to other types of cells. So we're going to start looking at the bacteria cells and the different structures associated with them. We're going to start with the glycocalyces. This is substances on the outside of the cell, so we're basically working on the outside, working in. And it's can be a kind of like a sticky, gelatinous type of substance surrounding the outside of the cell. So it's outside of the cell wall. Typically, it is composed of either polysaccharides, polypeptides, or a combination of those two. There's two different types of glycocalyces. There's capsule and then there's slime layer. The capsule tends to be very firmly attached to that cell surface, and it's an advantage to the bacteria to have a capsule, especially if it is a pathogenic organism. Pathogenic means that it's going to cause disease. And what we have found is that you can have two bacteria, same species, so more or less identical. One has a capsule, one does not. If it has a capsule, that capsule is going to give extra protection to the bacteria, and it's harder for a host to be able to recognize that bacteria if it's able to infiltrate and penetrate inside that organism. And so if it has a capsule, it tends to be more pathogenic. That's good for the bacteria because it increases its survival rate. It's bad for you. And that's one thing you're going to find in microbiology. When we often talk about things, we look at it from both sides. Look at it from you as a patient who may possibly end up with an infection and then maybe a full-blown disease. But we also look at things from the side of the microorganism. And what's good for one is not necessarily good for the other. So a capsule is not good for you as a patient, but it's good for the bacteria because it increases its chances of survival. It increases its chances of evading your immune system. Like I say, that's good for it, not for you. The slime layer is very loosely attached to the cell surface, and it's going to be water soluble, so it can be easily removed. It helps to allow the prokaryotic cell to be able to attach and stick to surfaces. Excuse me. So this is a picture showing, and picture A is a capsule, and in B is the slime layer. Flagella is another type of external structure, and not all bacteria will have these. Some will, some won't. Depends on the species. Sometimes within a species, it depends on the strain, like with the capsule. Flagella, if it has it, it's going to help move the bacteria. It's a very long structure. It extends, once again, we're talking about externally, so it extends beyond the surface of the cell. So it's going to help with locomotion. It's composed of three main parts: a filament, a hook, and a basal body. The basal body is what is going to help anchor or attach that filament and the hook to the cell wall. And so in this picture, you can see overall that you have extending an A extending out from the cell wall. You've got the filament. You have that hook that's helping to attach it, and then that basal body is what's embedded within the cell wall. Now, if a bacteria has a flagella, and remember not all of them do, it's going to have a certain type of structure and orientation to it relative to the overall cell, and that can be used to help you for identification of the bacteria. Some will have multiple flagella surrounding all over the cell, not just on one end, and it's not just one, it's multiple flagella on the sides, on both ends, and that is referred to as peritrichous flagella. Some may have a single flagella just on one end. Some may have multiple flagella, but they all originate from the same spot, and it's usually just on the end, and you may have like 2 or 3 extending from one spot. So if it's one flagella on the end, it's called monotrichous. You can have different terms for this. Amphitrichous is where you have just one flagella, but on each pole, on each end. So you have a total of 2, but they're just on each end. Peritrichous, as I said, is surrounding the entire cell. And when you have that tuft, so one spot, but the tuft of them coming out, that's lophotrichous. Usually associated or talked about with flagella is also what we call the axial filament, and this is where it's similar to flagella, and that but you have a filament that wraps around the cell, and so when it tends to contract, it causes the bacteria to look like a corkscrew, as seen in A. And the way it will contract, it gives kind of this corkscrew movement to it as it moves ahead. The function is to help move the bacteria through whatever environment it is. The bacteria is going to move in response to different stimuli. It doesn't move in a straight line per se. It has 2 different types of movements that we call run and tumbles. In this diagram, it's kind of showing the motion of a bacterium, how it starts off, as you can see in the upper left, and it kind of tumbles around and then it will move straight. That's called a run. It's when it's moving in a definite direction, and then it kind of tumbles and rotates around, and then it'll move again. I always say bacteria never took geometry, so they don't know that the shortest distance between 2 points is a straight line. They don't know to go straight from A to B. So in this drawing, it doesn't know to go from the upper left corner to the upper right corner. It's going instead to take the scenic route. So it's going to run a little bit one direction and then go, well, let me tumble and now kind of take a left-hand turn and move this direction and tumble. They're kind of interesting to watch if you're able to look at a living cell under a microscope. You definitely see it's not straight lines. Some additional external structures that can be found on bacteria are the fimbriae and the pili. These are both going to be much shorter than the flagella. The fimbriae are kind of sticky projections. They help bacteria to stick to one another and also to stick to surfaces. Sometimes you will find bacteria going to grow in an environment where there's liquid, and to keep them from being washed away, it's helpful to have these fimbriae to help stick to a surface. And this is showing the very long strands or flagella. Some cells may have both. And then the fimbriae are the shorter strands that are allowing it to stick to the surface. The pili, they are actually a specialized form of the fimbriae. They're also known as the conjugation pili. They're not as long as the flagella. Usually they'll only have one or two of these. Only certain types of cells will have these, typically only found in gram-negative bacteria, but not all gram-negative bacteria have them. The purpose of them is to help transfer DNA from one cell to another. So they're hollow, and here you can see the pili where it has extended from one cell to another. It's very controlled as to, it can't transfer DNA to just any cell. It has to be compatible. Essentially the same species, and even within that it has to be compatible. And this pili, essentially think of it as like a hollow connecting tube, and the DNA passes inside of it from the host cell to the recipient cell. Bacteria, the majority of them do have a cell wall. There are a few that don't, but the majority of them do have a cell wall. It helps to provide the shape and structure for the cell. It helps to protect the cell. Sometimes the cell wall can help the cell itself to attach to other cells. The cell can protect it and help resist the effects of antibiotics. Oftentimes with new antibiotics, that's one thing we can do is develop an antibiotic to target the cell wall of the bacteria. The cell wall is what gives the characteristic shapes to the bacteria, so obviously it's very important. The bacteria cell wall is composed of peptidoglycan. And with bacteria, we have been able to, for classification purposes, separate them into one of two groups, a gram-positive and a gram-negative. And the differences between these has to do with the composition of the cell wall and basically how many layers of peptidoglycan do they have. So this is showing an example. Like I said, the cell wall helps to determine the shape of the bacteria, whether they are spherical or whether they're rod-shaped. Now, this is showing a comparison of in A is the structure of glucose, and then in B is the structure of what is known as N-acetylglucosamine, otherwise known as NAG, and then also N-acetylmuronic acid, which is also known as NAM. That you have that basic template of the glucose and then add additional molecules to make the larger compounds from that. So when we look at the structure of what is this peptidoglycan that I've mentioned, that is what comprises the cell wall for bacteria. What it is these chains. You have these long chains. The chains are made up of alternating the NAG and NAM components that we just saw, the N-acetylglucosamine, N-acetylmuronic acid. So they alternate in there. So you have these long parallel chains. Connecting the chains is a small little chain of amino acids. Now, if you have multiple layers, how do you connect the layers? Well, then you're going to have what we call a cross bridge. It's a tetrapeptide, which means it has 4 amino acids, and it's a cross bridge that then connects one layer to the next. So as you can see, it kind of looks like scaffolding, this lattice work in here. And that is what the peptidoglycan looks like. When you look at a gram-positive bacterial cell wall, you notice it has a very thick layer of the peptidoglycan. So you have multiple layers, as we just previously saw, so it looks like a thick scaffolding. There are some chemicals in there that are called teichoic acids. When you do the gram stain, it is what we call a differential stain. That means you use 2 different stains or 2 different dyes. And so you're going to be able to just visually look at it and distinguish between things. The gram-positive bacteria are going to look purple. So in this diagram, you can see in A, you have a gram-positive cell wall. And if you look at a cross-section of that enlarged up, you can see you have on the very bottom the cell membrane, that cytoplasmic membrane or plasma membrane, it's the same thing. All cells have that. It's that phospholipid bilayer. You have proteins embedded in it. That's what those little purple things are. And then there's a little space, and then you have, as you can see, a thick layer of peptidoglycan. It's multiple layers thick. You've got the thick scaffolding in there. Embedded within it, you have these teichoic acids. Some of them are teichoic acids. A lipoteichoic acid just means you put a lipid on top of or attached to that acid. So those are embedded within the peptidoglycan. Notice the thickness of that peptidoglycan layer. When you look at a gram-negative bacteria, the peptidoglycan layer is going to be much thinner. It's only about 1 or 2 layers thick. And then there is an additional layer, membrane, on the outside of the peptidoglycan. So from the inside of the cell, you're going to have the cell membrane, you're going to have a thin layer of peptidoglycan, and then after that, you're going to have another membrane that's referred to as your lipopolysaccharide layer. Embedded in this is what we call Lipid A. Lipid A is something that is going to be released upon cell death. This does become a problem, or at least something you need to be aware of whenever you are treating someone with a gram-negative infection. And the reason for that, and we will get into this later when we talk about diseases, etc., is that lipid A that's in this lipopolysaccharide, when it is released upon death of the bacteria, it can cause different symptoms with the patient. It can induce fever, it can induce vasodilation, it can induce the inflammatory response, and this can all lead to shock. It can have blood clotting. So the reason why you need to be aware of this is that if you have a gram-negative infection, you want to clear up the infection. You want to kill basically the gram-negative bacteria. But do you want to kill them all at once? No, you don't, because you would have this sudden release of the lipid A, and that can lead to shock in your patient. And so I know it sounds strange, but you want to do more of a controlled, regulated killing of the gram-negatives. So you just need to be aware of that. When you do the gram stain, the gram-negative bacteria are going to look pink or pinkish red. So when you look at the schematic drawing of the gram-negative, you have that cytoplasmic membrane. Notice how thin that peptidoglycan layer is as compared to the gram-positive. Much, much thinner. And then you've got this whole extra outer membrane layer that has the lipopolysaccharides in it, has the lipid A in it. There are proteins that are in it. This is a whole layer that's not there in the gram-positive. That's why they react differently to the gram stain. This is also going to play a role as to why they may react differently to antibiotics. As I said, there are a few bacteria that do not have a cell wall at all. They're usually very small. For years, oftentimes they were mistakenly identified as viruses instead. However, they do have ribosomes, viruses do not, and so they are classified now as bacteria. With that plasma membrane or cytoplasmic membrane or cell membrane, it's all the same thing. It is a phospholipid bilayer. So you do have these phospholipids in there, but there are also going to be proteins that will be embedded within the lipids. Some of these are peripheral proteins, meaning they extend only on one side, and some of them are integral proteins. An integral protein extends all the way through the width of the membrane, so they're sticking on the inside of the cell, but they're also, the other end is sticking out on the outer surface of the membrane. We refer to it as a fluid mosaic model. And what that just means is that you do have some side-to-side motion of the components of the membrane. It's not like it's cemented and set and it can't move and this is where I'm sitting. No, there is side-to-side motion. Kind of think like, I don't know, maybe a little seaweed, you know, when you're on the coast and you see something moving in the water. There's some back and forth. Not a whole lot, but there's some back and forth. And this shows in this schematic drawing the phospholipids. If you remember, if you've studied the chemistry, that the phospholipid has the hydrophobic tails, fatty acid tails, which in here are drawn as or colored as yellow. And then you have the phosphate head, which is hydrophilic. That's the phosphate group, and it's shown as blue here. It's a bilayer, meaning you have two layers. So those fatty acid tails that are hydrophobic are going to point towards each other. And then in this drawing, you have the proteins that are colored as purple. As you can see, there are several of the integral proteins that are extending through. And then you have peripheral proteins as well. For the membrane, there is some energy storage there. It can help for photosynthetic bacteria. It helps to absorb the light. It is selectively permeable. That means it regulates what can pass through that membrane. It is not a free-for-all. Only certain things can move in, only certain things can move out. And it's going to regulate and very tightly control the movement of things across the membrane. This will help to maintain proper concentration gradients as well as electrical gradients. Basically, if it cannot regulate and properly control the concentration of things inside the cell versus outside the cell is going to die. There is a slight charge with cells. They are more negative on the inside and slightly positive on the outside. We can actually measure the electrical potential, which is known as the resting potential of the cell. It's around minus 70 millivolts. Sodium, which has a positive charge, tends to be a higher concentration outside the cell. Things like chlorine, which are a negative charge, tend to be higher concentration inside the cell. And so that's how you end up with a net negative inside and positive outside. Some of the function of the membranes to help with the movement of things across the membrane. There's different ways of moving things. Initially, more broadly classified as passive versus active processes. Of the passive processes, you have diffusion, you have facilitate, diffusion, and osmosis. Passive process does not require any energy. You are going to be moving down the concentration gradient. In other words, you're going to be moving from an area of high concentration to an area of low concentration. That's the way everything wants to flow anyway. And so it's not going to cost you any energy to do so. So, what are the different types? So we said diffusion, facilitate, diffusion, and osmosis. Diffusion is just simply when a substance moves across that membrane and it goes from high concentration to low concentration. Usually this is going to involve very small chemicals, those that are lipid soluble, because to pass through that fatty acid portion in the middle, where it's yellow in this diagram, that's hydrophobic. Things that are hydrophilic, that easily dissolve in water, are going to have problems. And so those that are hydrophilic, they will often pass through some of these proteins that are embedded, act as channels. Think of like somebody stuck a straw in and there's a pore in the middle, and in the middle things that are hydrophilic now can easily pass through. And facilitate diffusion, you're still moving from an area of high concentration to low concentration, but you need the help of one of these proteins. And then osmosis is essentially the diffusion of water across the membrane. Osmosis is one thing to keep in mind. You're talking about the movement of water, and obviously in water you usually have things dissolved in it. So to get an equal concentration of water does not necessarily mean an equal volume. Just keep that in mind. When we talk about osmosis, we usually talk about what we refer to as tonicity, of which there are 3 different types. You talk about a comparison of what is dissolved in a solution as compared to what concentration of things are inside the cell. So you're looking at what is a solute, which is what is a substance that is dissolved in a solvent. Usually when we're looking at cells, water is the solvent. And then the solute is what gets the component that gets dissolved. It may be salt, it may be sugar. In summertime, your water would be your solvent, and your lemonade powder would be the solute, or your Kool-Aid would be the solute, whatever. And so with tonicity, what you're looking at is a comparison. We have 3 different types of solutions. So with an isotonic solution, just break down what the word means. Iso means equal. So that means the concentration of the solute inside the cell is equal to that outside the cell. So because outside the cell it's equal to inside, you say that solution is isotonic to the cell. You have as much, now you're doing 2 things. You're looking at the concentration of the solute, but what you're concerned about is the movement of water. So you have, because the solute concentration is the same, you have equal amounts of water moving out. That's what's moving in. And in this diagram, it is showing on the upper portion cells that do not have a wall, versus on the bottom, those type of cells that do have a cell wall. So this is a normal situation for most type of cells, is to be isotonic. In column B, where you have a hypertonic solution, hyper means more, so you're doing a comparison. The solution is hypertonic to the cell. So that means the solution has more solute than what's inside the cell. Now, with osmosis, what we're saying is diffusion of water. Yes, we're looking at the concentration of the solute. The solute, we're not worried about where the solute's going. It may not be able to pass across the membrane. What we are concerned about is the water. To try to equalize the concentration of water, if the solute concentration is high, the water concentration is low. So think of like a lemonade glass. If the cell, inside the cell is like a glass of lemonade, you have an 8-ounce glass and you put 1 tablespoon of powdered lemonade in there. If outside the cell, you still have an 8-ounce glass of water, but now you put 3 tablespoons of powdered lemonade in there. The concentration of the lemonade is higher, which means the concentration of water is lower. Water tends to flow towards the salt. towards the solute. And so in this case, in a hypertonic solution, when you place a cell into a hypertonic solution, the water is going to flow out. Now, if water is flowing out of the cell, especially in a cell that does not have a cell wall, it's going to start to shrivel up. Now, because you have a cell wall that's helping to protect you some, so you can withstand the effects a little bit better than if you did not have a cell wall. In column C, you have a hypotonic solution. That means hypo means less or below. So the solution has less solute than inside the cell. Like I said, water is going to want to flow towards the higher solute, so water is going to flow into the cell. Now, as you can see in the upper picture, if water is flowing in a cell that has no cell wall, just has the membrane, and water keeps flowing in, and water keeps flowing in, that cell is swelling. It can only swell up so much. It's just like you when you're blowing up a balloon. You're blowing it up, it's getting bigger and bigger. It can only go so much before it pops. And that's what's going to happen. Now, in those cells, such as bacteria, fungi, plants that have a cell wall, that cell wall is giving extra protection, preventing it from bursting. And so having this cell wall does provide a little bit of extra protection for these changes in the environment of the osmotic pressures. So the cell can withstand these changes a little bit better. Active process is, there's two types, active transport and group translocation. It's going to depend on the size as to which one of these happens. Excuse me. With active transport, you can have what's known as uniport, where you're moving one substance in one direction. You can have what's known as antiport, where you're moving two substances and you're moving them in opposite directions. So one comes in and one goes out. And then coupled transport is basically where you're pumping a combination of things. You may be pumping something out, and then it's going to come right back in again. Symport is when you're pumping two things in the same direction. Antiport is two things but in opposite directions. Group translocation is when you have a larger substance that's too big to pass across the membrane, and so you're going to have to move it through specialized proteins. Now, active transport, you are moving things against the concentration gradient, so that is going to cost you energy. This is a summary table of the different processes for transporting things across the membrane of bacteria. If we move into the cell, you've got cytoplasm. The cytosol is the liquid portion. It's mostly water. It does have, in a particular area referred to as the nucleoid, the DNA in there. It may also have inclusions, not all cells, but some may have inclusions, which is where you have different chemical deposits. This is an example of some inclusions in one particular bacteria. Some bacteria may have endospores, not all do, but some will. It's a way of surviving unfavorable environmental conditions. So a vegetative cell is a cell, as you normally would think of it, it's actively growing. And when there are unfavorable conditions, environmental conditions, maybe temperature changes, maybe pH changes, maybe suddenly nutrients are running out, whatever the condition may be, it's going to trigger that vegetative cell to form endospores. And the endospores, the vegetative cell may eventually die, but the endospores will survive. They're resistant to very extreme conditions like heat, chemicals, radiation. And just so you know, we often use the endospores as a guide as how well various sterilization techniques, how effective they are. It's kind of like, the best thing is if it can kill the endospores. Some things we do may kill vegetative cells, but not the endospores. So we use this as a guide as to how effective various, say, chemicals are. The formation of the endospore, first, before anything happens, you have to replicate that DNA, make another copy of it. So you've got a copy of the DNA that's going to survive. Then the cytoplasmic membrane tends to start to kind of pinch in a bit, and it forms what is known as a forespore. The cell's going to be continuing to grow, and then as you can see, that forespore is kind of enclosed now within a second membrane. The vegetative cell, now its DNA tends to disintegrate and the cell's dying. You have now left the endospore. You have a coat that tends to form around it, which is known as a spore coat. You have the outer coat. So you have multiple layers here, and eventually it's released from that original vegetative cell, which is previously died now. But that DNA is protected by multiple layers, and so that's why it's able to survive these harsh conditions. When favorable conditions resume, then that cell is, the spore basically germinates and you get a new vegetative cell. The prokaryotes, remember, do not have membrane-bound organelles, but they do have non-membrane-bound organelles. So they do have ribosomes. This is where protein synthesis will occur. One thing I just want to say is the ribosomes that are found in prokaryotic cells are slightly smaller than those found in eukaryotic cells. There is also cytoskeleton. There's different protein fibers that are extended throughout the cell. They help to play a role in determining the shape of the cell. They do play a role in cell division, and sometimes it can help with movement through the environment. So in this fluorescent stain, you can see the glowing, that it's a stain that's added that penetrates through the cell and stains the cytoskeleton. So you can see it kind of looks helical in shape. For the archaea, some of the external structures, there are glycocalyces also that are found that helps to kind of help them form biofilms, where the cells stick to each other and then they also stick to other surfaces. Some of them may also have flagella, and just like we saw in the bacteria, where you have the basal body, the hook, and the filament. They do have fimbriae, and some of them have another type of structure similar to the fimbriae that's called a hammy, which has these, it looks like a fishing hook is what it looks like on it, as you can see here. Most of the archaeas do have a cell wall. However, they do not have peptidoglycan in the cell wall. So that's one way you can distinguish between the archaea and the bacteria. They do have a cytoplasmic membrane. Once again, that's going to help maintain the gradient, the chemical gradients. It's going to help control what's moving in and out of the cell. You have different shapes associated with the archaea, more unique shapes than you see with bacteria. So here you can see quite diversity of shapes. The cytoplasmic characteristics are similar to the bacteria. They do have, the ribosomes are the same as those as the bacteria. They do have a cytoskeleton. They do have DNA, and that's something I have not mentioned. The bacterial DNA is circular, so is the DNA in archaeas. Eukaryotic DNA is linear. Some of the differences between the archaeas and the bacteria, besides the cell wall, but when we look at the cytoplasm, there's additional differences. Although they do have the 70S ribosomes, they do have some additional ribosomal proteins. They have some different enzymes that they use to make their RNA. Their genetic code is more closely similar to that of the eukaryotes. So this is a nice summary table showing the differences between the archaeas and the bacteria. Remember, both of these are prokaryotes. When we move on to look at the eukaryotic cells, the glycocalyces is not nearly as organized as like the capsules that we see with the prokaryotes. There are some depending on the type of cell, some glycocalyces, it may help to anchor animal cells to each other. It can help strengthen that cell surface. Sometimes it's there to help prevent against dehydration, and part of it is used to help with recognition, basically communication between the cells. Fungi, algae, plants, and some of the protozoans will have cell walls. Animals do not. The cell wall, what is it composed of? Well, it depends on what you are talking about. Plant cell walls are composed mostly of cellulose. Fungal cell walls will have some cellulose, but it's mostly chitin. And then algae cell walls are going to be composed of other polysaccharides. So this is showing with a eukaryotic cell wall. Now, once again, all eukaryotic cells, as I've said before, all cells will have that cytoplasmic membrane. It's going to be the fluid mosaic with the phospholipids and the embedded proteins in it. There will be things like cholesterol to help maintain the structure of it. That will be embedded within the phospholipids. It's going to be, once again, the functions controlling what's moving in and out of the cell. So there are similarities like that. This is showing using a transmission electron microscope. It's showing in detail what the cytoplasmic membrane is looking like. There are different ways that cells can move and take in substances. This is showing endocytosis, which is a way of moving things into a cell. So you can see here, you have the amoeba and the pseudopods, so these arms, and this is often with protozoans, the way that they're going to move. In the upper left, it notices it looks like a paramecium that's unfortunately going by, and the pseudopods, those extensions of the cytoplasm, it's kind of like they flow out, and they're starting to reach around in the upper right picture. And now in the lower right picture, it has almost completely engulfed that paramecium, and by the lower left, it is completely engulfed now inside, and the amoeba, and it's going to, basically, that just became dinner. It's going to break it down and utilize anything from it as nutrients. So this type of active transport, because you're moving things in large substances. Endocytosis, as we just saw, is bringing something into the cell. We don't see this with bacteria or the archaeas, but we do see it with the eukaryotes. It's a way of bringing very large substances into the cell. You can't, in that previous picture, you can move that paramecium across the membrane. It's way too big. Phagocytosis is essentially like cell eating, so that's what we saw. Penocytosis is with fluid. Exocytosis is the opposite, where you're taking waste products inside the cell and expelling it or secreting it, pushing it out of the cell. Some eukaryotes will have flagella. The structure, the arrangement of it is different than what we saw with the prokaryotic cells. There is a filament, there is a basal body, but there's no hook. Usually it's going to be found just on one end of a cell. They tend to move in a rhythmic fashion and are used to help move the substance or the cell through the environment. And this is just showing a depiction of what it looks like. So as it moves, it's going to propel the cell through the environment. Cilia are going to be much shorter. They're usually more numerous, though, than what we see with flagella. And as they move, it's helping to move fluid across the surface of the cell, as you can see here. The cilia are much shorter and, as I said, much more numerous. So as they're moving, they can help move the organism through its environment. Or if it's stationary, it can help as the cilia are moving. It's kind of like seaweed moving once again, and it propels fluid across the surface of the cell. Eukaryotes are also going to have some of these non-membranous bound organelles, such as the ribosomes. Now, the ribosomes found in eukaryotes are going to be slightly larger than those in prokaryotes. In prokaryotic organisms, the ribosomes, we refer to them as 70S. It has to do with the size. And in eukaryotes, it's 80S. The subunits, they don't add, just so you know the size of them, you don't add the numbers together up like you would normally do in math. So you have a 40S and a 60S, but it ends up being an 80S. You also have a cytoskeleton, this network of fibers and tubules that helps to anchor the organelles. It helps for movement of substances through the cytoplasm. It helps to, once again, protect the shape of the cell. And as you can see here, there's different types of tubules. Microtubules, microfilaments, intermediate filaments, they differ in size that are found in the as part of the cytoskeleton. You also, in some cells, have centrioles and centrosomes. These are going to play a role in mitosis with the cell division. They're not in all of your eukaryotic cells. And this shows examples of what they look like. In terms of the membrane-bound organelles, eukaryotes, certainly you have a nucleus. That is going to contain the DNA. It's the largest organelle in the cell typically. There is a semi-liquid portion inside called the nucleoplasm. You do have, excuse me, in it the nucleolus. And this nucleus, this membrane that surrounds it, is called the nuclear envelope. It does contain pores in it, which are small openings. Basically, it allows for communication with the rest of the cell. So as you can see here, in both the TEM, remember, is the transmission electron micrograph, the picture allowing you to see cross-section. So you can see that there are these small openings or pores. Like I said, that allows for communication. The endoplasmic reticulum, which we abbreviate as ER, is this whole network of these hollow, flattened tubules. They're continuous with the nuclear membrane and then will extend out into the cell from there. It does act as a transport system. There's two types of endoplasmic reticulum. There's this, excuse me, smooth ER and the rough ER. As you can see here, the picture depicting them, the smooth ER, as you see, the surface looks very smooth. There's nothing attached to it. The rough ER has, it looks like little bumps on it. These are ribosomes that are attached to it. The smooth ER is functioning oftentimes to help make various lipids and has specialized function in specialized cells. The rough ER has those membrane-bound or ribosomes on them that's going to be involved with protein synthesis. The Golgi body or the Golgi apparatus, this is where typically it will receive large molecules and it has processing before those molecules are exported out of the cell. One of the main things that it's going to be receiving are proteins. So proteins that are made on the rough ER, on those ribosomes, typically from the rough ER, excuse me, will transport in a vesicle, a little small sac, if you will, and be transported to the Golgi body, where there will be further modifications made. Maybe you're going to stick a sugar on the end of it. Maybe stick a little fat on the end of it. You're going to make some modification to it. And then you secrete it or export it out of the cell from that point on. They look like a stack of flattened sacs. Typically, the incoming vesicle comes on one end, and then it fuses with it, moves down layer by layer until the end it breaks off and then is exported out of the cell. I often describe it as looking like a stack of pancakes, and it looks like you've poured syrup on the top, and it's going to dribble on down the sides. So this is what it looks like. So the vesicles arrive on one end, in this case showing coming from the rough ER. It has the protein inside of it, the newly made protein, and then you're going to modify it. And by the time, think of it like that stack of pancakes, by the time that syrup drips all the way through and comes out on the bottom, it is now complete. All the modifications are complete. It's ready to be exported. You have additional membrane-bound organelles like lysosomes, peroxisomes, vacuoles, vesicles. These are often storing various chemicals or transferring chemicals within the cell. They may store extra nutrients. The lysosomes are often referred to as the recycling center because they would contain very highly digestive enzymes that will break down anything that it comes in contact with. The peroxisomes contain enzymes that can help break down various waste products. This is showing an example of a vacuole, the central vacuole that is seen in plant cells. As you can see, it's a large, very large component within the cell. It's a storage facility. And this is showing an example of endocytosis where you are taking in, specifically phagocytosis, a solid substance. In this case, the cell is engulfing a bacteria. It encloses it within this vesicle. Basically, part of the cell wall surrounds it, or the cell membrane surrounds it, forming this vesicle or the sac, takes it in. Now, a lysosome, remember those have the highly digestive enzymes, will fuse with that vesicle, releasing the enzymes, breaking down the bacteria. The cell's very, very good about recycling things. So any components like proteins from the bacteria, it's going to break those down. You reuse the amino acids, reuse the nucleotides from the DNA. Anything that it can use, it will reuse. And then any of the waste products, as you can see, that vesicle is going to be the remains. Those waste products will be secreted, eliminated from the cell. The mitochondria is a double membraned organelle, so it has two layers of it. This is the powerhouse of the cell. This is where most of the ATP is going to be produced. Something interesting about the mitochondria is that in the internal matrix of it, it does contain 70S ribosomes, and it does contain circular DNA. If you remember, that is what is found in prokaryotic cells, not eukaryotic cells. But we're talking about eukaryotic cells right now. So this is showing a cross-section of the mitochondria. You can see where it has the two layers. That inner layer membrane has quite a bit of invaginations, or it's not smooth, straight surface. It has all that infolding. That's increasing the surface area. Whenever you see any type of that infolding like that, increasing the surface area, it tells you that something very, very important must be occurring here to allow increased surface area, to allow increased process occurring here. The chloroplast. Some eukaryotic cells, such as plants, will have chloroplasts. This is going to be able to absorb solar energy, the sunlight, and carry out the process of photosynthesis where it's producing ATP. The chloroplast also has a double membrane like the mitochondria does. It also has DNA in it, which is circular. It also has 70S ribosomes in it. And here you can see the relative structure of the chloroplast. And in this table, it is a summary showing the comparison of prokaryotes and the eukaryotes and the various organelles that are present in there. The endosymbiotic theory is something that comes up when we talk about eukaryotes versus prokaryotes. The eukaryotes are newer from an evolutionary standpoint than the prokaryotes. Prokaryotes were here first. Prokaryotes are smaller, eukaryotes are larger. Now, something that has come up deals with the mitochondria and the chloroplast in an effort to try to explain Why do both of these organelles that are found in the eukaryotic cells, why do they have some of these components that are found in prokaryotic cells? They should not have the 70S ribosomes. They should not have circular DNA. Those are only in prokaryotes, so why are they there? And this is a way of trying to explain this. What the endosymbiotic theory essentially says is that the mitochondria and the chloroplasts were at some point way back in time probably independent prokaryotic cells that were ingested by the new eukaryotic cells. And that what happened was, say for the mitochondria, a eukaryotic cell engulfed it, and it became a symbiotic relationship. And it was beneficial to both. Why? Because if the mitochondria was initially a prokaryotic cell and now it's inside another larger cell, it's protected from the environment this way. And how is the eukaryotic cell protected or benefited from this? Well, if the mitochondria or the chloroplast, both of them are producing ATP, that's energy. So you now have a supply of energy that you didn't have before. And the chloroplast and the mitochondria are protected. So this is what the endosymbiotic theory is. Most people, most scientists do accept this now, though not everyone does. And overall, here is a comparison of the archaea, the bacteria, and the eukaryotes, comparison of the various components that are found from the extracellular, like flagella, cilia, to the cell wall, the plasma membrane, versus then what is inside the cell.